DNA Replication by Kaushik Bukuri and Noah Otto. So DNA is a very, very large molecule. And if proteins were to start at the top of the strand and work their way through the end, it would take a very, very long time, and it would also be very inefficient. So to speed up this process, proteins break up the replication of the DNA strand into many parts. Specifically, they start at particular sites, called origins of replication, and these are basically a short stretch of the DNA that has a specific sequence of nucleotides. And so here, proteins attach the DNA to start the process of replication. So here on the screen, we see a replication bubble, and this is the place where those aforementioned proteins are attached to the DNA strand. And this red boxed area right here is the replication fork, and this is where the parental strands of DNA get unwound. But before we can actually replicate the DNA, the double-stranded molecule has to be sort of unzipped into two single strands, as you can see here in the middle. And this is where the enzyme helicase comes into place. So that enzyme is this cyan box right here. And helicase is an enzyme that disrupts the hydrogen bonding between the base pairs to separate the strands as shown. So this enzyme just goes through the DNA and separates the two strands. After these strands are broken up, single-strand binding proteins, so those are these magenta pink diamonds here, so those are single-strand binding proteins, and they attach to these singular strands here, and that prevents them from rejoining again. Unfortunately, all of this unraveling causes lots of stress on the double helix ahead. Fortunately, the help of the enzyme topoimerase, which is this green oval right here, that comes in handy here, and this enzyme ensures that those stressed strands unwind and rejoin to prevent tangling so that the next steps in replication can proceed smoothly. Now we can look at the actual process of replication, and for this we will look at the enzyme primase, which is this purple triangle here. Primase takes these unwound sections of parental DNA strands and it starts a complementary RNA chain, adding matching RNA nucleotides one at a time. So you can see this gray thing is the RNA nucleotide that it is gradually adding. And so this primase adds the complementary RNA chain normally in 5 to 10 nucleotides at a time. However, it's important to note that each strand has directionality. So on this picture, the top strand has 5' on the left end, as you can see here, and this bottom strand has 3' on the left end. So clearly the directionality and orientation is flipped. So replication only progresses in the 5' to 3' direction. And as one would expect, replication is carried out in two different processes to accommodate these directional differences between the two strands. On the top strand, only one primer is needed, as you can see here. And this one primer is known as the leading strand. And on the bottom strand, a series of segments need to be synthesized since the enzyme is working away from this replication fork. And these small segments are known as lagging strands.
Okay, so where we left off, we had two strands. The first one, the leading strand, is orientated in the 3' to 5' direction, towards the replication fork. The other strand is orientated in the 5' to 3' direction, away from the replication fork, and that is called the lagging strands. As a result of their different orientations, the two strands are replicated differently. So for the leading strand, we have a DNA polymerase 3, which is the yellow rectangle I drew. This binds to the leading strand and then walks along it, adding new complementary nucleotide bases, A, C, G, and T, to the strand of DNA in the 5' to 3' direction. This sort of replication is called continuous. Now for the lagging strand, we have an RNA. We have a primase enzyme, which is the brown circle I drew, which makes numerous RNA primers, which is the red rectangles I drew, And these RNA primers bind at various points along the ligand strand. We also have chunks of DNA called Okazaki fragments, and those come immediately after the RNA primers. They are added to the ligand strand, also in that 5' to 3' direction. This type of replication is called discontinuous, as the Okazaki fragments will need to be joined up later. So next, once all the bases are matched up, A with T, C with G, an enzyme called exonuclease strips away the primers. This is done by our DNA polymerase 1, which is the orange circle I drew, and the gaps where the primers were are then filled by yet more complementary nucleotides. The new strand is proofread to make sure there are no mistakes in the new DNA sequence. And finally, an enzyme called DNA ligase, which is the blue rectangle-ish shape I made, that seals up the sequence of DNA into 2 continuous double strands. So at the end of this process of DNA replication, there are 2 DNA molecules consisting of 1 new and 1 old chain of nucleotides. And that's all.